All right, so we finished the first 4 weeks of embryonic development. Now it's time to talk a little bit about how we describe development in a clinical or medical sense, and the difference between abnormal development and normal development. And for this, what we really need is a vocabulary lesson. Bear with me. A lot of these terms are terms that we're going to use over and over again, so it's worth knowing them. Now, the basic process here, morphogenesis, this is the term that your book uses to describe normal development. That's really all it is. Dysmorphogenesis is abnormal development. And while clinically, medically, we don't use the term dysmorphogenesis a great deal, the study of dysmorphology is the study of birth defects or congenital malformations. And then we often use the term dysmorphic, which is a descriptive term to describe somebody who appears to have abnormal development or an organ system that appears to have abnormal development. It's most commonly used in the term dysmorphic facial features. And so what's shown in this picture here, this is an illustration of dysmorphic facial features. This is a couple, the woman who has had normal development and the man, the husband, who has not. He has a condition called Treacher-Collins syndrome, which affects the development of the face and head, but nothing else. So intellectually, he is normal. Physically, he is otherwise normal, but he has significant changes to the development of his face and hearing loss as a result. And then this is their daughter who has the same condition he does. Treacher-Collins syndrome is a Mendelianly inherited disorder. It's autosomal dominant. Although she is more mildly affected, you can see her facial features are not quite as distinctive, although clearly there's something there. What's very interesting about this is that I actually found this family in an article. This is a British family that was written about their decision to have a child. So they had to go through IVF to have her, and they chose not to select against the genetic change that caused Treacher-Collins syndrome. And for that reason, they were written up in the newspaper because apparently some of their neighbors felt that they should have done that. So an interesting ethical debate there. But at any rate, an excellent example of dysmorphic facial features. What causes abnormal development? Well, any of a number of things. Genetic mutations for sure. Much of Treacher-Collins syndrome can be attributed to the TCOF1 gene, although there are other genes that can cause it as well. There are teratogens. These are environmental agents that alter a developmental process. Alcohol is the example that's given in your book, although there are many, many others. Positional causes. This is basically the position the baby gets to in the womb, and the classic example of a positional birth defect is clubfoot. So clubfoot occurs when the foot gets fixed into an orientation that it can't move out of. So if you think about your foot, you can move it in any of a number of directions, but if it were stuck in one orientation, you couldn't. And if a baby happens to be in that position in the womb, where I might point out it does get fairly crowded eventually, for too long a period of time, then the muscles and underlying tissues may sort of grow into that orientation, and then the baby might not be able to move the foot. It's a positional birth defect. Multifactorial causes, any combination of the above. So birth defects can be a combination of genetic, environmental, and positional factors. And then a lot of them really fall under who knows. A lot of times there is no cause, you never figure it out. And that's just kind of how it goes. Okay, different types of birth defects. And it is worth noting that birth defects are actually relatively common. About 3 to 5% of all babies will have one, and many of them will be minor and very correctable, or in fact, maybe not even in need of correction, but some, of course, will be more severe. So your book highlights the difference between a malformation and a deformation. Basically, this is the distinction between what's causing the birth defect. So a malformation is a birth defect caused by an anomaly intrinsic to the developmental process that produces that particular body part. Okay, so the example there is spina bifida. With spina bifida, you have a failure of those neural folds to close the neural groove. Okay, an opening is left, and that produces spina bifida. The birth defect there is resulting directly from an anomaly of the development of that part of the body. A deformation is different because in this case you've got an external force acting on the developmental process that then produces the birth defect. And again, positional birth defects like clubfoot are an excellent cause. Clubfoot is not caused because the musculature or the bone structure of the foot is abnormal. It's caused because the position of the baby acting on the development of those structures produces the birth defect. And that's an example of club feet for people who aren't familiar with what it looks like. Other ways to describe birth defects: isolated birth defects, a birth defect that occurs by itself with no other birth defects. Isolated doesn't tell you anything about its cause, just that it occurs by itself. A sporadic birth defect is one where we don't know what's causing it. All right, it's not inherited. It doesn't appear to be part of a syndrome, so sporadic birth defects are usually isolated birth defects. And we really don't expect the recurrence risk to be terribly high for future children in the family. A familial birth defect is one that's running in the family. It may be isolated, it may not be, but it's not sporadic. And so this is an example of a family, actually a Maori family, with inherited clubfoot. And the way this particular pedigree is keyed, the filled in squares are bilateral club feet, and if it's half filled in, it's unilateral. It is interesting to note that in this family, it does appear that only males are affected. I suspect one of the primary reasons for that is that there are just an awful lot of males in this family. But it is worth pointing out that birth defects are, as a whole, much more common in males than in females. Any number of theories as to why that is. But it does seem like males are more susceptible to birth defects, particularly sporadic birth defects. Okay, the next distinction to make here is syndrome versus sequence. So a syndrome is a group of anomalies that all occur together and share a single direct cause, okay? Like a genetic disorder, a genetic mutation that produces cleft lip, heart defect, and club feet. All of those caused by the one genetic change. And of course, it doesn't have to be genetic, it could be environmental. A great example of that is achondroplasia. This is the most common inherited form of dwarfism, or genetic form of dwarfism. And what you're seeing in the two pictures here is a brother and sister. The sister has achondroplasia, the brother does not, and you can see over time the change in their relative heights, although the brother is in fact younger than the sister. Achondroplasia is characterized by short limbs, although relatively normal-sized trunk, frontal bossing, which is a little bit of a protrusion of the forehead, and a low-set nasal bridge. So if you feel your nose, most people have a bone in their nose that sort of produces this straight line along the bridge of the nose. If you have a low nasal bridge, then instead of that straight line, you've got a bit of a divot or depression there, and the nasal bone is not as large, and so you don't get that straight line. Achondroplasia is caused by mutations in the FGFR3 gene on chromosome 4. And it is worth pointing out, since we're going to cover lots of birth defects and lots of conditions that produce significant challenges to people leading what most people would consider a normal life, they don't have to do that. I think most of you will recognize this fellow. This is Peter Dinklage. He is one of the stars of Game of Thrones. He has achondroplasia, and he has certainly been very successful. Not every birth defect or syndrome or sequence or what have you means that you can't have a great life. All right, sequence. A sequence is a rarer situation than a syndrome. A sequence is more of a snowball effect. So in this case, you've got a group of anomalies that share an ultimate cause, although they have a very different immediate cause, okay? And this is the snowball effect. You have one thing that happens that produces the next thing, that produces the next thing, that produces the next thing. And the best example of that is oligohydramnios or Potter sequence. So this is basically not enough amniotic fluid. And the way this usually works, or often works, is a baby that's born with renal agenesis, not born who's developing with renal agenesis, meaning absence of both kidneys. If you don't have kidneys, you can't produce urine. If you don't produce urine, you don't have amniotic fluid. That's basically what it is in the later stages of pregnancy. If you don't have amniotic fluid, you can't breathe it into your lungs and cause your lungs to develop. If you don't have amniotic fluid, you can't move. If you can't move, then your limbs become contracted, fixed into their positions, because the baby needs to be able to move them in order to develop normally. And if you're squished in there because you have no amniotic fluid, then you get this sort of compressed, flattened look to the face of the rest of your body. So in this case, renal agenesis doesn't cause contractures of the limb and poor lung development, but renal agenesis causes absence of urine, causes absence of amniotic fluid, which causes these other issues. Snowball effect. Okay, so just to sort of drive home the point about how we categorize birth defects and highlight the idea that actually the same birth defect can have lots of different etiologies, we'll go back to club feet. So this was our first one. These were sporadic club feet. In this particular baby, there was no other cause found, and they were likely positional, the way the baby was sitting in the uterus. So they're isolated, they're sporadic. It's a deformation because they were positional. Then this baby has club feet that are caused by a genetic condition called mucolipidosis type 2. It's a Mendelian disease. So in this case, they're a malformation. They're caused by the genetic mutation that the baby had, and they're syndromic because the baby has other issues related to that single genetic mutation. This just illustrates the point that not every syndrome is genetic. This baby has syndromic club feet caused by prenatal exposure to Zika virus. So not genetic, but still a syndromic club feet, still a malformation. And this highlights the idea that not every set of birth defects fits nicely into our classification scheme. So this is a baby with arthrogryposis, which is a congenital contracture disorder. Contractures just meaning that the joints are fixed in positions they shouldn't be fixed in. That includes the joints of the ankle and produces club feet, but in this baby, other joints are affected as well. For this baby, they never did find the cause. They looked at genetic changes, exposures, nothing was ever found. The arthrogryposis was isolated, so while there were multiple joints affected, there weren't any other issues going on. So it's hard to say if it was a malformation or a deformation, if it was truly syndromic or not. Hard to tell. Okay, now your book does spend some time on animal models. Not really the focus of this class because we're really targeting medical concepts that can be used primarily for genetic counselors. But it is helpful to understand the parallels in development across species and how animal models have contributed to our understanding of human development. So I did sort of recreate the list of animal models that's in your book. You guys are probably familiar with many of them anyway. And I did want to point out that when picking models, there are a couple of factors that are important. You certainly want them to be similar to humans, although of course nothing is a perfect match, and sometimes you take a similarity in a particular organ system or genetic pathway. They have to be easy to care for. You like to have a short generation time, which means that they make their babies relatively quickly. It allows you to conduct experiments over a short term rather than long term. And the same thing with lifespan, although whether you want it to be long or short kind of depends on your research, but you might target lifespan as a particular factor. And it is important that they not be terribly sympathetic creatures. There is a reason we don't do research on puppies. Okay, so signaling pathways. Your book also spends some time on this, and I do encourage you to go back and look at all of the pathways in the book and familiarize yourself with them. They will come back up in various other lectures, although it's a little difficult to anchor them now since we don't have the developing organ systems to talk about. But to give you the basic idea, cell signaling is at the core of embryonic development. The basic idea is that within a cell, gene expression alters. It leads the cell to produce a signal that it then expresses outside of the cell itself. Interaction with that signal triggers neighboring cells to alter their gene expression, and then they may change their cell signaling, which alters the next cells, and on and on and on, and it cascades, generally producing different types of development in different areas based on how genes are expressed and which signaling molecules are being released. The genes that are involved in the embryonic cell signaling pathways are often highly conserved across species, meaning that the gene involved in this process in a fly is probably quite similar to the one in a rat and the one in a human. Okay? And a great example of this are the Hox genes. So the Hox gene family is a huge gene family. They're all transcription factors. Transcription factors are genes that transcribe DNA into RNA, so they control gene expression, clearly critical for embryonic development. And the Hox gene family is highly involved in the regulation of body patterning. So how do we make the normal body plan? They are, of course, highly conserved across species, and they occur in clusters. And it's kind of interesting how that happens. So if you look at this picture here, the embryo on the top is a fruit fly, and then underneath it are listed its Hox genes. And then the next embryo is a mouse, which at this stage is really quite similar to a human. And then you have all of the mouse Hox genes. And of course, the mouse has many more than the fruit fly. But if you look at their color coding, you can see that many of the mouse's Hox genes are directly derived from those in the fruit fly. Because again, they're highly conserved. But in the mouse, over time, in the progression from fruit fly to mammal, we've seen duplication of many of those Hox genes and development of new functions for the duplicates that lead to greater complexities in body pattern. Okay, so how does this relate to human disease? So this table is a list of many of the different diseases that can be caused by Hox genes in humans. And so I'm going to highlight this one. So this is HOXB1, causing hereditary congenital facial paresis type 3. It's a recessive disease causing facial palsy. It's an inability to control the muscles of the face, hearing loss, and unusual facial features, dysmorphic facial features. So you can see it's caused by this HOXB1, but that gene is expressed in roughly the same region in the mouse or human as it is in the fly, and in the fly it's controlled by the labial gene from which the HOXB1 gene is derived. And if you look at a fly with a labial gene mutation, it also has anomalies of the face and mouth and eyes in some way similar to what you see in the humans. So all these things are linked. And again, definitely familiarize yourself with the gene pathways in the book. They're going to come up later, and we'll try to anchor them in the organ systems in which they occur when we get there.